This instrument is called ELISpot, and this has been built by CTL. Normally, or like even now, there is a technique called ELISA, which is enzyme-linked immune sorbent assay. But ELISA is more like a semi-quantitative technique where you know that something is being secreted and you can just measure those concentrations. But we never knew that which cells are secreting those proteins or those antigens. Then came in the picture this ELISpot, which was more like an ELISA, but on a solid surface. So we do the ELISA, but using a membrane where cells are plated. And then when cells are secreting a protein or an antigen, we capture it using enzyme, and then a colorimetric reaction takes place. And then those spots will be forming. We also call them like SFU or spot forming units. That's how you count them. And you will know that out of 300,000, even if one cell is secreting something, you're going to catch it using ELISpot. So it's that precise. ELISpot technique has been really important in terms of knowing the specific cell responses, like out of the whole sample, if certain cells are responsive and others are not, like especially in vaccine development. We wanted to know if it's just the antibody response that's going to be there, or what should be the exact method to develop a vaccine. We knew that antibody responses were waning in a few months, but then the cells, they were still reactive and responsive when they were stimulated again using part of SARS-CoV virus cells again secreted those parts. So, and also like in drug development, this technique is very important. Also in clinical trials, when there is longitudinal trial, you want to see that how a certain response is changing over the time. You can really, in control settings, ELISpot can be really very informative about those cytokines or those proteins to form. ELISpot technique is like one of the most important pieces that ELISpot has applied or has answered was, say that even when developing a vaccine, which part of a virus should be used to make the optimal vaccine. So you can use different antigens from a vaccine, and if you are stimulating cells with that, ELISpot assay can show that against what epitope or what part of a virus cells were most responsive, but then proactive response or protective response versus non-protective response, or the cells are going to maintain that immune response in long term against a certain epitope, or they are going to lose, then we need additional stimuli for that. But now this machine is almost fully automated. It has like really high throughput visual like images, and then also the counting is automated. You can stack multiple plates at the same time and then just hit command and you go out of the way, and then it keeps counting them, each plate one by one, like, and then it's really fast. And so you can do 96 well plate in a few minutes. It's like really fast in terms of counting. And you can do more than, like I said, like now kits are available that you can do 3 molecules at the same time on the same cell in the well. So you get more answers in the same cell. And this is like really sensitive technique. Like with this technique, even a few hundred, like one cell in 300,000 cells, if it has a response, you will see the spot there and you will know that it was there.